Now, here is something I am going to tell you, absolutely new. Focus on this. The blood that is flowing through the lungs. Let us look at only one RBC, forget about one RBC. Millions are there, let us look at one RBC. Where is it coming from? It is coming from the heart. Okay, from the right ventricle it is going towards. Now, is it compulsory that that RBC will flow through an alveolus and will have an opportunity to get oxygenated? No, no. If that RBC happens to pass via a bronchi or a trachea, okay, or some smooth muscle tissue which is lining the trachea, is it getting an opportunity to pass through alveoli? No. Does it get oxygenated? No. It is passing through like some other tissue. It is not going through. It is not going through that part of the lungs where it will have an opportunity to oxygenate. Are you getting the argument? Well, there is this after all there are major blood vessels in the lungs. They have their walls. They have their smooth muscles. They need oxygen, and that RBC is not the RBCs which flows through those organs is not going through alveoli. So, it is not getting oxygenated. So, what happens to that is a very interesting question we will try to answer. The heart is somewhere here, do not bother about it, but from the heart comes a very important blood vessel which you know already, it is aorta. What is it? Aorta. It is the same guy which shows compliance. Hello, this is aorta. Okay. From the aorta, and then of course you know these are the carotids which go to the which go where? Carotid which go through the through what? Through your neck to your brain. But can you see author has drawn some two very small vessels and they are going to what? They are going to the trachea and the bronchi. What about these vessels? Now let us see these vessels. Okay, so this here the dark blue indicates oxygenated blood and the light blue indicates deoxygenated blood. So let us talk about the blood that is being pumped by the left ventricle. Where does it go? It goes in the aorta. But some of it goes via these vessels, it goes to the bronchial circulation. Where does it go? Bronchial. Is it alveolar circulation? No, it is a part of the lung. But is it alveolar? No, it is part of what? Bronchial. It is a small component, but it goes. So it goes to the bronchial circulation. Are you okay so far? Okay. Now, in the blood in the bronchial circulation, it goes into two channels. Some of them go into what you call as the deep bronchial veins, and which go where? Follow the story. It is very interesting. Follow the story. Let us go to the right ventricle. Right ventricle. What kind of blood? Deoxygenated blood. Are you okay? From here, the blood will go into the pulmonary arteries. Deoxygenated blood. It goes to the lungs. Fully oxygenated. Fully oxygenated. Just follow the story. It comes out there and it is joined by, before it goes to the heart, blood passing through the lungs, fully oxygenated. What is the target? Now go to the heart. Left auricle, left ventricle and go everywhere in the body. Are you with me? There it receives some amount of deoxygenated blood. Get the point? And what is this deoxygenated blood? That blood has already given. Is it oxygenated? No. Why? Because it has flown through the bronchi part, where it was not the alveoli and where it did not get oxygenated. Okay. Therefore, we have the blood from the bronchial circulation. So, this connection shows that the blood is coming from the deeper part of the lungs, not the alveoli. The upper is coming from the alveoli and then they mix together and going to the left ventricle and then everywhere. Am I making sense there? So, okay, so let me put it this way. You draw the sample from the pulmonary vein, oxygenated pulmonary vein, just as it is coming from the lungs, okay. And what do you find? Partial pressure is 104. How much is the partial pressure? 104, okay. Now you take another sample from the same vessel as it is ready to enter into the heart. Take the sample there and you find that the partial pressure is about 96, 95, 96. There is a drop. Why is the drop? It's a mixture. So, actually what goes to the heart is not fully, is not the blood that is fully saturated, there is a slight drop, okay. And we call that blood as the shunt blood, okay. So, let us see. Blood comes from the aorta. Now, the blood that is going from the aorta, some of it go bronchial, whereas some of it goes here, some of it goes, just follow another, what do you call it as? Hilar bronchial vein, it goes to vena cava. So, that is our traditional route. So, what are we doing just now? We are trying to find out how the body deals with the blood supply to that part of the lungs which do not have alveoli and which do not have the opportunity to get oxygenated. Okay, and that. Okay. Now, this is very well seen.
Sir,
yes, please.
There can't be. Is there any rationale behind this particular thing?
The rationale is simple. The rationale is that after all, that part of the lungs.
No, which both could have gotten.
Which one?
To vena cava, if you go to the previous slide.
No, I think anatomically this might be a better way to let the blood go there. After all, the quantity is very little. Okay, that will. I think it is the design of the anatomy. The system evolved that this is It may be a more circuitous route to take it there. I don't know. Okay, I am just guessing. Okay, so here we have the blood that is coming into the lungs. What is the partial pressure? 40. Now it has gone into the alveoli. What is the partial pressure? 104. Okay, what is the oxygen partial pressure? 104. Okay. So whatever oxygen has got into the blood, I will call it partial pressure of oxygen 104. Okay. Now 104, and then it is coming, and then can you tell me why this drop is happening from 104? It is going to about say 96, 95, 96. Why is the drop? We did just now. The bronchial, okay, the blood that has irrigated bronchial, and then it goes to, so then it will go to the heart, and then from to everywhere else, and then it goes to different tissues. It again falls back to 40. This point is very clear, and we call this system as, this blood flow is called as what? Shunt flow. It's called as shunt flow. Okay, so now let us see what happens to the blood. The blood is going from there into the left atria, left ventricle, aorta. It has gone to different tissues. Let us take any tissue. Let us take pancreas, liver, whatever. And we are now look, as once the blood is pumped from the heart, it goes through major arteries, arterioles. There is hardly any change in the blood partial pressure of oxygen. So from the heart, if it is coming 95, 95, 95, it's still 95. By the time it goes to arterioles, it is still 95. It hasn't dropped. Then it finally goes to meta arterioles and capillaries. If we have capillaries, it is still what? So the partial pressure here is almost same as the partial pressure of blood if you retrieve it from the left ventricle. 95 mmHg, same, it goes same. But the moment it comes into the capillary, now where we are in a capillary, and these are the target cells where the blood is going to supply its oxygen. And in the tissues, what might be the partial pressure there? 23. Now 23 is an average value. It can be depending on the physiological state of the cell. It can be 5, it can be 10, it can be 15, 20, 30, whatever. Okay, but it is, so so Guyton has given the averages of how much? 23 mmHg is the partial pressure. Extracellularly, and as it comes, extracellular may be little more, maybe 40 mmHg. Now the blood arrives into the capillary. Okay, now the walls are suddenly very thin. Okay, and there is an RBC going, and in the RBC there is hemoglobin, and that hemoglobin molecule is fully saturated. It is only having it's almost having 95 mmHg pressure. Outside the capillary there is fluid which has 40 mmHg, and there is immediately there is a cell with 23 mmHg. Huge gradient. Are you getting the argument? As a result of huge gradient, the hemoglobin will immediately. That's the property of hemoglobin. It will lose its oxygen. Okay, and lose its oxygen, and by the time the blood goes from here to here, which is a distance of about 1 mm. Okay, and then the partial pressure is reduced to how much? 40 mmHg. Okay, now it is in the veins, and then that's what will be carried to the vena cava and back to the heart. Okay, all right. Now,
sir,
yes.
Is it different in the fluid outside of the cells and inside of the cell?
Because there is a gradient. This is a gradient. Okay, so the blood is steadily flowing. Okay, so as it is in the cell that there are mitochondria, and those mitochondria are continuously using oxygen. So there is a gradient. It's maintained. You have to maintain. That's our most important part of homeostasis, is to maintain that gradient. Okay, that is very well. Your argument is fully answered in this question. What we are just understand, it's a very nice diagram. You will enjoy looking at this diagram. we are looking at partial pressure of oxygens at different levels, right from outside. Okay, outside is how much? Look at the first one, room air, our air around us. If it is dry, what is the partial pressure there? You are okay with this? Yesterday we okay. Then from there you go into it goes into the trachea. As it goes into the trachea, it mixes with the air that is already there. Okay, which means it drops to where? 149, and then from there it goes where? In the alveoli. In the alveoli is about how much? 100, okay, whatever it is, 100, 100, whatever. And then it goes into the arterial blood. I mean, there now it is that mixture. Okay, now it has been reduced. The author tells us it is about how much? 90, 92. Okay, then it goes into the it will give away to the tissues, and then it will reduce to what? 40. Now this is the most important part. What is the partial pressure of oxygen in the mitochondria? 2. Mitochondria, actually they say that even 1 or 2 mmHg of partial pressure, mitochondria is happy. Okay, that's what it needs. Okay, but it can't do less than that. I mean, it has to be provided. So actually we have this entire system, okay, geared over a process of evolution over millions of years, given the fact that air around us is 160 mmHg and your target is to take it to a mitochondria at that with that much of partial pressure of oxygen. That is how the entire system is geared. That depends as to how much of hemoglobin, what is its affinity, at what rate heart is pumping, all that. Ultimately our aim is one. What is the aim? To make sure that mitochondria have a very steady, and the environment of the mitochondria has a partial pressure of about how much? 1, 2 or 3. 2 or 3, that's enough. But for maintaining this, you need that. Okay. Okay, you can read about. Okay, we are talking about the other side. Other side means what? Let's talk about. That's only one function. The other function. Okay. Please remember, hemoglobin also has a very interesting role in transport of carbon dioxide. Okay, carbon dioxide. We'll talk about carbon dioxide. I'll come to those details. So we are looking. I'm sure you are familiar with the earlier part, alveolus. But in this particular part, the author tells us something different. In the alveolus, what is the partial pressure of carbon dioxide, please? 40 mmHg. How much it is? This carbon dioxide. And the blood that is the blood that is deoxygenated blood that is coming from the heart. What is the partial pressure of carbon dioxide there? 45. So it's 45 here. Okay, and it is 40 there. Okay, so there is gradient as a result of that, and you keep on tracking it, and you find that by the time you are done almost 1/3 the distance of the capillary, the CO2 is gone. And not gone means it has come down from 45 to 40. Okay, and as the blood goes, it will remain 40. So the partial pressure of carbon dioxide goes on oscillating between 40 and 45. By the time you go to the tissues, you go to 45, then you again go back to the lungs, get rid of that 5 extra, come back to 40, and this is how it keeps on operating. So how does it work in the tissues? Again, blood is coming, oxygenated blood. It has what partial pressure of carbon dioxide is there? 40 mmHg. In the tissues it is how much? 46, outside is 45. This gradient is good enough. It will pick up the carbon dioxide, and then as a result of PCO2 40 will become how much? 45, and that is what will be carried to the heart, to the lungs for getting rid of that carbon dioxide. Let us look at this very amazing molecule. Again, back to the structure of hemoglobin. It is in quick steps. Guyton tells us about some of the. There is two molecules of succinyl CoA glycine. They come together, and this molecule we will call as what, please? Pyrrole. Good. 4 pyrroles come together to give rise to what? Protoporphyrin 9. Okay. Then if you in protoporphyrin ring gets an iron molecule, okay, it becomes what? Heme. That is what the molecule is. Okay, and what do you call it as? Protoporphyrin ring. You call it as, and it's like a coin. You know, it's flat. That molecule is flat, and well, these are not amino acids. Okay, they are not amino. No, this is not amino acid. This is not a protein. This is not a protein. Okay, this group. What group? This group. This heme group. Okay. This is the heme group, and this heme group then goes and sits at the center of a large protein molecule, okay, and then it becomes hemoglobin. The protein molecule itself is globin. Okay, it combines with the heme. Heme consists of what? Protoporphyrin, at the center of which there is an iron atom. Are you okay so far? Okay, and if you look at the structure of this molecule, author tells us that there are 1, 2, 3, 4 N's are there, nitrogens are there, part of that molecule, and they hold the iron molecule in a position. Okay, and then to go ahead with that, this protein molecule. Okay, okay. How many protein molecules are there? There are 4 protein molecules. How many are there? 4 protein molecules. Okay, it's a tetramer. 2 of them are called alpha 1, alpha 2, beta 1, beta 2. 4 of them are there. Okay. Now each one of them has a 1, 2, 3, 4. Can you see the 4 heme groups there? And each heme group is capable of binding to one molecule of oxygen, O2. So this molecule can carry one molecule of hemoglobin, can carry how many molecules of oxygen? 4. Okay, and when it binds to 4, we will say that the molecule is saturated. Okay, and that is what should happen when the RBC with its hemoglobin is travelling where? Through the lungs, where there is an opportunity for the molecule to pick up the oxygen. Okay. Now, can you see the okay, let's appreciate this image. These are the protoporphyrin ring is there. Can you see the iron at the center? Okay. Now there is a huge protein molecule. Okay, and that has those ribbons. You understand the protein molecules are ribbons and cords. This thing. There are several alpha helices, and they are labeled as A, B, C, D, whatever. And one of the helix is called as F. And that helix has an amino acid histidine. Hello, histidine is an amino acid. That histidine, okay, it binds to iron. Means what am I talking about? I am talking about how the porphyrin ring is coming in contact with the protein molecule. How is it coming? In the protein molecule there is histidine. There are it's a huge it's a quaternary structure, that hemoglobin molecule. It has at a particular place the histidine molecule. For that you will have to go into the chemistry of hemoglobin, which we are not doing, and that histidine molecule is directly bound to iron. Okay. This is of great relevance, and I will come to that a little shortly. And look at the 2 states in which the molecule can shift. When there is no oxygen, it can take oxygen, and then when it takes oxygen, it looks like this. This is called as oxygenation. What is it called as? Oxygenation. Okay, and Guyton takes a point very clear. It is not oxidation. It is not. It is oxygenation. It's a very loose bond. Okay, okay. It can readily, and it's reversible. So if oxygen is there, it will loosely bind. Okay, and if the oxygen is not available, it will just go away. So it can readily shift between 2 different states. The molecule can readily slow. Okay, the reaction is very rapid, requiring less than 0.01 seconds. Read the bottom line. It's interesting. Approximately 25 trillion RBCs are there, and what? Each one RBC is packed with how much of hemoglobin molecules? Can you read for me? Huge amount. Huge amount. We're talking about packed. That's packed with RBCs. Okay. Now this is you can't really understand hemoglobin molecule unless you go through the process of how the hemoglobin molecule keeps on changing depending on the amount of partial pressure, or in simple language depending on how much of oxygen it is carrying, and what are the different stages that influence the affinity of oxygen for the hemoglobin. Let me try to clarify my point. First of all, there is a very small percentage, 2-3% of oxygen can go through the blood in a dissolved form. Hemoglobin has a role to do that? No, hemoglobin has no role to play in that, but that is only low 2-3%. Okay? About 98-97% of the oxygen that is being carried through the blood is bound to hemoglobin, because hemoglobin greatly increases the capability. I mean 30 times, 40 times, 50 times, it is huge. We really can't do without hemoglobin, without the capability. Okay, all right. So, go to the top curve, and let me go to the next one to really explain to you what the story is. Let us see there. You have to understand this graph, you have to, do not miss the point. I will try to make it as simple as possible. Very simple. On the x axis here, what do we have? Partial pressure of oxygen, okay, partial pressure of oxygen. So, let us take a look at the blood that is just coming out of the lungs. So, if you are there, then I would say that you are here. Am I right there? Hello? Hello? Are you okay? We are here, okay. And when you are here, and supposing you are having 100 ml of blood, I think did I do that or I did not do that? I missed this slide, let us do it, because without this you can't go there. Some basic properties of hemoglobin, okay, or blood. 100 ml of blood, go to the second point there. 100 ml of blood contains how much? 15 grams. Just separate, hemoglobin will be 15 grams. Okay. 1 gram of hemoglobin binds with about 1.34 ml of oxygen. 1 gram. 1 gram contains how much? You can carry how much oxygen? You have to remember these figures. So, 100 ml of blood with 15 grams of hemoglobin can carry about 20 ml of oxygen. You just have to remember these numbers. Okay. So, under healthy conditions, 100 gram of blood contains 50 grams of hemoglobin, can carry 20 ml of oxygen. Done? Oxygen I am talking about. Good, good, good. This is expressed as 20 volumes percent. That is the way the physiology people use the language. What is 20 volumes percent? It means that 100 ml of blood with equipped with 15 grams of hemoglobin will carry how much of oxygen? 20 ml. Therefore, 20%. Okay. All right, and then now with this bit of information, let us move here. So, if we are here, so let us collect 100 ml of blood that has just been through lungs. Are you okay with me? 100 that is just come. Because it has just come from lungs, we presume that it is oxygenated and its partial pressure is about here. Okay. Then it should carry what volume of oxygen? 20 ml. Okay. And given that all those that 100 ml of blood that I am having in front of me, that 100 ml of blood, presuming that all its RBCs, in those RBCs all the hemoglobin molecules, each hemoglobin molecule has 4 sites for oxygen, they are all occupied. Therefore, I will go there and I will say that the saturation is 100%. Have we been able to coordinate 3 very important things? In oxygen in terms of volume, okay, when it is fully, okay. Saturation means make sure that all the sites are occupied, okay. And when that is the scenario, then what is the partial pressure? 100 mmHg, okay. Now, I will ask a very interesting question. What happens if the partial pressure of oxygen, I am not taking it from lungs, I am taking it from somewhere else, and it is not 100, it is 80. Are you with me? That is the question I am asking. If it is not 100, then if it is 80, then what is the status of other parameters? Means what? Now, I am not here, okay, I am here. In that particular case, then if I am here, then what is the amount of oxygen I am still having? Maybe 19, hello? It is little less, is it 20 now? It is not 20, it is little less than that, and then you can go from this point, you can go to saturation. Or if I go now to 40, why will I go to 40? I will go to 40 because, tell me why I am going to 40? It is in the tissue. Okay. When I am going to the tissue, so this is the blood. Now I am collecting the blood that is come. Okay, let me put it this way. I am collecting the blood from vena cava. Okay, you are okay so far? Vena cava. That blood should have partial pressure of 40 mmHg. So if I take that blood still has how much of volume of oxygen? 15. Isn't it extremely revealing that the blood that is going to the tissue has 20 ml, the blood is coming is still 15. It's a lot. This is a lot. Why do you think nature has margin? Absolutely, one word, absolutely correct answer. It is the margin. Margin means if you suddenly start working, that 15 may go down to 14, 13, 12, 10, okay, but you still have a lot of oxygen. Okay. So, we call this as the oxygen dissociation curve. what do you call it as? Oxygen dissociation curve. And the thing that you readily note and that defies our common sense is that the line is not a straight line. Okay? It is a sigmoid line. Okay? Now, why is that line a sigmoid line is another interesting question, and let us try to. So, it still has same, it has about 15 ml of blood as it is as we are going to the. Again, I will take you to my favorite place. My favorite place is Leh. I have never been there, but still. Okay. Now, try to keep on try to appreciate the beauty of the molecule. That's what we are doing. Look at the molecule and clap. What a great molecule it is. So, the is a pressure of oxygen in the blood. Okay. This is on the x axis. Is it possible, I mean, when I am at sea level, okay, we are somewhere here. As far as alveoli is concerned, where are we? As we are somewhere here at sea level. When you go to Leh, you are not here, but you are here. Do you agree with me? Because it is the oxygen is less there, and I am not able to get what I am good. It's rarefied. So I don't get it. So I'm somewhere here. Even when I am here, I mean this is a, I mean it's a massive, it's a drop, okay, from 100 to 80, okay, 20% drop is there in terms of what? Availability of oxygen from here to Leh. But if I were to do that, how much of oxygen is still there if per 100 ml of my blood? 18. So from 20 to 18 is not a great drop. Are you getting the point as to why the nature has come up with this particular dissociation curve, where with such a huge difference with reference to from here to there, but in some years 18 is still good. Okay, it's not 15, it's not 12, it's just 18. Okay, so again that is the advantage of having, had it been a straight curve, you would not have had that advantage. Okay, so the nature has come up with this. Now, why this property is? So we are now looking at the normal pressure, normal arterial. We are somewhere around 100, and normal venous is 40. When you are doing exercise, you actually go here. Somewhere venous blood, you fall, you are not 40, you are less, okay, but there is enough buffer or enough margin so as to give you the affinity, follow the point, the affinity of hemoglobin to oxygen is dependent on the pH of the blood. On what? pH of the blood. And this is extremely important, I will tell you why. Yet another secret of hemoglobin. It is very meaningful. I will tell you why. You are doing exercise, rigorous exercise. As a result of that, your skeletal muscles are generating huge amount of carbon dioxide. As a result of that, the carbon dioxide can combine with water and give rise to carbonic acid. That carbonic acid can ionize and give rise to H ions. Those H ions, where is this happening in your muscles? Those H ions can lower the pH of your blood slightly. When the pH of the blood is lower slightly, the affinity for hemoglobin, the curve is moved to the right, which means the affinity of hemoglobin for oxygen is reduced. Hello, are you getting it? Let us see, you are here somewhere or you are here somewhere, go straight. Okay. Now, there are three, the middle one is the normal. What is the pH there in the middle? 7.4, normal. But let us take an extreme, you are doing great amount of exercise. As a result of H ion, the pH of the blood has fallen to 7.2. It never really happens, but let us consider for understanding. It has fallen where? 7.2. Now, at the pH 7.2, as the blood is flowing through the skeletal muscle, what is happening to the affinity? What is happening to the affinity which was here? What is happening to the affinity of the blood for oxygen? Is reduced. It is what? Reduced means what? It will liberate the oxygen more readily. It makes sense. Why does it make sense? Because the muscles need more oxygen. Are you with me? It's very interesting. Okay. So, depending on the need at a given place, the hemoglobin decides whether I am going to increase my affinity for oxygen or decrease my oxygen, and exactly opposite happens when the same blood flows through the lungs. Okay. The pH again goes up. When the pH goes up, okay, its affinity for oxygen goes up, and but in the lungs there is abundance of oxygen. So, it can combine with the oxygen more rapidly. So, look at again keep on looking at the hemoglobin molecule. When it is going through the lungs, okay, because now are there so many H ions now? No, those H ions they have gone to, okay, what I will tell you what happens to that. Again the carbonic acid is again converted into water and carbon dioxide is released. Once carbon dioxide is released, okay, the number of H ions goes down, so the pH moves upward. Once the pH moves upward, then this curve moves in this direction, and hemoglobin has more affinity for oxygen. Now, I am going to show you a very interesting picture. I don't want you to read anything, but just look at the pictures. And do you identify this guy on the right? Who is he? Who is he? Who is he? Still nobody. Half of you don't know who is. Who is he? I am talking of his dad. Okay, Christian Bohr. What is his name? Christian Bohr. What is his name? Christian Bohr. This response of hemoglobin to reduce the affinity of the hemoglobin to oxygen depending on the pH is called as Bohr's effect. What is it called as? Bohr effect. Bohr effect. What do you call it as? Bohr effect. Read the bottom line at the bottom. What does it say? This is called the Bohr effect. Okay, and you are not going to confuse what Bohr is. I have told you the entire relationship. Okay, so you know Bohr. Okay, he was a medical person. Christian Bohr was a medical person, and Niels Bohr we all know. Okay, so got it? Very clear. Don't forget it. Okay, that's why the history of science is interesting. Okay, all right.